All right, I'm going to go ahead and kick us off here. The people joining us seems to have slowed down a little bit, so I think it's a good time to kick off. My name is John Downey, and I have the pleasure to serve as the treasurer of the Dystonia Medical Research Foundation. I personally have DYT1 dystonia, which is why I'm so excited to be here tonight as the moderator. And like many people with DYT1 dystonia, I started to present symptoms when I was still a kid. And I have many family members who also have DYT1 dystonia. My family was one of the many that ended up participating in the study that led to this discovery. And this discovery of the DYT1 gene was a big deal for myself and my family. I went on to get deep brain stimulation to help me with treat my dystonia in my 20s, and it has been tremendously successful for me personally. Now, I also have two dystonia-free children that would not have been possible without this discovery. So I'd like to welcome everybody again for joining us and let you know that this is being recorded and it should be available next week on the DMRF website. The format tonight is intended to be conversational, and we'd like to hear your questions. So please send your questions via the chat, and we will get to as many as we can. We've already received many questions in advance, and I'm looking forward to the conversation. So let's go ahead and get started. Tonight, I'm honored to be joined by Dr. Xander Brakefield, Gloria Zulius, and Nipa Patel. Each of them have a very impressive resume, and I'd like to go ahead and do a quick bio for them. Dr. Xander Brakefield is a professor of neurology and neuroscience program at Harvard Medical School and a geneticist in the neurology and radiology services at Massachusetts General Hospital in Boston. Her research focuses on understanding the molecular etiology of movement disorders, including early onset dystonia, as well as gene and drug therapies for neurologic diseases. Dr. Brakefield has received numerous awards, and all very impressive, and serves on the board of directors for the American Society for Gene Therapy and on NINDS study section. She's a former member of the Dystonia Foundation Medical Scientific Advisory Council and is a longtime friend of the DMRF. Dr. Brakefield currently receives funding from the DMRF for her work normalizing DYT1 cholinergic neurons by CRISPR disruption of mutant Tor1A allele. I'm going to take a little bit of a breather after reading that mouthful of a paper title. Dr. Zulius is an internationally recognized expert on genetics of movement disorders, particularly hereditary dystonias and Parkinson's disease, and has identified a number of other dystonia genes, including DYT6 and DYT12 genes for inherited dystonias. Dr. Zulius has identified a founder mutation in the LRK gene that is responsible for 13% of Parkinson's disease among Ashkenazi Jewish individuals. In her lab at Massachusetts General in Harvard, is currently working on finding genes involved in the penetrance of DYT1 dystonia and LRK2 Parkinson's disease, defining the mutational and clinical spectrum associated with various dystonia genes, understanding the genetics of XTP, and identifying novel genes and risk factors for later onset focal forms of dystonia and Ashkenazi Jewish Parkinson's disease using next generation sequencing of whole exomes and genomes, as well as genome-wide association studies. Dr. Zulius has also served on the DMRF's Medical and Scientific Advisory Council and has been a longtime friend of the DMRF, serving as faculty for many scientific workshops and as a member of the faculty for the Sam Bellsberg 6th International Dystonia Symposium, which is scheduled for June of 2023 in Dublin. Dr. Nipa Patel is a physician and the director of Movement Disorder Interventional Program in the Department of Neurological Sciences at Rush University in Chicago. Dr. Patel is interested in improving the quality and delivery of care for patients receiving deep brain stimulation and education to improve the utilization of new therapies in movement disorders. In addition to DBS as a treatment for dystonia, Parkinson's disease, and other movement disorders, Dr. Patel is also exploring the use of MRI-guided focal ultrasound to manage movement disorders. Dr. Patel is a member of the Movement Disorder Society, the Huntington Study Group, Parkinson's Study Group, and American Academy of Neurology. She was a DMRF clinical fellow while doing her movement disorder fellowship at Baylor University, and Dr. Patel has also been a longtime friend of the DMRF, speaking at many educational meetings and webinars. So again, I'd like to welcome you all and thank you for being here for this important discussion and allow me to, I also want to first kind of thank you all for the work that you're doing. As someone who has benefited personally from the work, I just want to send out a heartfelt thank you. And with that, I think we can go and move into the bulk of the program. We want to make sure that make sure that everybody's on the same page. And so with that, kind of starting with definitions is usually a great place to start. And so Dr. Patel, I'd like to start with you. And can you just tell us a little bit, what is DYT1 dystonia? Frame it up for us.
All right, so DYT1 dystonia is a form of familial dystonia where the gene, I don't have to get into the details of it, but the gene has been associated with causing dystonia. And in the description of all of my esteemed colleagues, there's a couple of words that I think really helped us better understand this condition. So DYT dystonia is heterogeneous. That means that one gene can look differently in different individuals of the same family and differently amongst other people who are not related to each other but carry the same gene. And penetrance is also really variable in this condition. And penetrance means, well, some people could have young onset dystonia starting at age 5 or 6. Others could have it as teenagers, which is the majority of the patients that have DYT1. But it has been described in older individuals as old as in their 30s and 40s. I don't believe it's been described in people at 70 and 80 years old as the first presentation, but it can affect people at various different stages and phases of their life, and it can look differently. Majority of the patients are affected in more than one body region, so their head, their neck, their arms, their legs, their body. But some people may only have a focal dystonia where it's affecting just their head and neck, or maybe an arm or a leg. And with the discovery of this gene, we've learned a few different things. One, just how differently one gene can work in different individuals. But two, also learned a lot about strategies to help treat it. And one of which is deep brain stimulation, which works quite effectively in this gene group.
Great, thank you.
Yeah.
So a question, we're here tonight to kind of talk about, we're on the 25th anniversary of the discovery of this gene, which was, foundational work. And, Dr. Brakefield, Dr. Azulis, you were there. So I kind of want, talk to us about like what went in, and how to like kind of talk to us about like, put us back there, like what went into the identification of this gene. We'll start with you, Dr. Brakefield.
Yeah, well, I can, with me, it goes back to the late 1970s. And I got a call from John Menkes. The Bellsbergs had just, with some other families, had formed this foundation to study the disease. And he was just trying to recruit people to study it. So he called me up and he said, would you be willing to submit a grant application to get funding to study this disease? And I had never heard of the disease. So I said, well, let me go down to the library and do some research, and I'll let you know if it fits into, what I'm studying. And it did. And so I wrote a grant, and I said, but I really need money quickly because I'm about to lose somebody. I need to, get their salary. And if they work on this, if I write this grant, could I get the money within a month? And he said, yeah, if we like it, you could get it within a month. Anyway, our first proposal was to study different enzymes in cells from the patients. And I have to say, right from the beginning, that, the patients were always heroic in donating not only blood samples, but skin biopsies. We do fibroblasts out of them. So we had quite a large stash of different things to study. But anyway, after the first couple of years, I remember I went to Canada where the Bellsbergs lived, and I said, we looked at everything we could in these cells, and we can't find anything different. I'm sorry, it doesn't work. And he said, and I said, so I won't be applying for another application. He said, no, you're just the kind of person we want. You're so honest. He said, just find another strategy. So, and that's where I moved then to Boston, and Lori Roselius had just graduated from Brown University. It was her first job. And I said, let's try this new strategy. This, you had just come out, and it's called linkage analysis. Lori may try to explain it to you, but it was definitely a new technology. And she was on board. She said, okay, let's try it, and we applied for funding from NINDS, which is the National Institutes of Health. And they said, there's no way you could find this gene because there's so many types of dystonia. You'll never be able to, find one gene that causes any particular type. And that, but the DMRF said, yeah, give it a try, because they were really desperate. And we met some of the families. Lori was wonderful at meeting the patients and dealing with them. And we just started slugging away at trying to find this gene. Basically, most of the time there was just 3 or 4 of us. And it was really, I call it grueling work. It was mostly grueling for Lori. Yeah.
Yeah.
Yeah, so as Andrew said, we use something called linkage analysis and positional cloning, which is like old school, very old school now. This was way before the Human Genome Project, way before we even had like maps of what genes were on what chromosomes, so we had like no knowledge. But what we did actually have was a good clinical team in Stan Fahn and Susan Bresman, who knew dystonia very well and could phenotype, very well. And so what they did was they went through their dystonia patients and they tried to figure out which ones had a very similar clinical presentation. And so then they told us these are the families with the most similar clinical presentation. And so those are the families that we concentrated on. And there was also a group that were Ashkenazi Jewish, so that there was a potential for a founder mutation among the Ashkenazi Jews. And so we had some very large non-Jewish families with like 7, 8, 9, 10 affected members. And then we had much smaller Ashkenazi Jewish families with 2 or 3 affecteds. But they all had the same clinical presentation, early onset, generalized, starting in the leg usually, sometimes the arm, and, over the course of 4 or 5 years generalizing. And so basically what linkage analysis is you just try to, you take pieces of DNA that show differences that are spread across the whole genome, and you basically test them all and you ask which ones segregate or travel together with the disease and which one and which ones aren't in the controls. And so you find a piece of DNA that has a variation that is always only seen in the affected people and not in the controls. And then because you know where that piece of DNA is located, what chromosome it's on, you then know that the gene is somewhere near that. And so that's the linkage analysis part. And then the positional cloning part is where you then try to actually get that DNA in your hands so you can figure out what genes are in there. So today, the positional cloning part is all done by the Human Genome Project, right? Because we can just go to the computer, say we're interested in this segment of a chromosome, and it'll chug out every single gene that's in there. And so what took us 5, 6, 7 years, you can do in like less than 5 minutes now. And so basically, that's what we used was linkage analysis and positional cloning to get to the disease gene using families with a very particular clinical phenotype or features.
Yeah,
I will.
that's really
I'll
good.
agree with Dr. Brayfield. That does sound very grueling.
Well,
Yeah.
I always tell people we went on for 10 years, I think, and every week we would meet, and I just put out a bottle of Tums and a bottle of aspirin, and just because it was just going to be painful to see how little progress we had made, although we kept making progress, but it was very, very slow and very tedious. But we couldn't give up because nobody else seemed to be interested. That was the other thing. At one point, I think one of our other diseases that we were looking at, we finally said we can't do more than one, and we gave all the samples to another researcher, but nobody else wanted to do it because it just was just hard work. And especially when you're in science, I mean, you're supposed to be publishing papers about exciting stuff, and here it was just, oh, now we're a little closer. Oh, now we figured out we're a little closer. Anyway.
No, yeah, it to me personally, it sounds kind of like science fiction, right? Like, fast forward, but you fast forward today, you say, you can, because of the Human Genome Project, this can be done much, much quicker. But, back when this work was happening, it predates all of that. Yeah, I can understand why it would take so long. One of the things I wanted to make sure that we touched on, and kind of talking about the identification of the gene is, what role did the DMRF play in this? And I know, Dr. Brayfield, you mentioned like the Bellsbergs, who are the founders of the foundation, reached out to you to kind of pull you in to work on dystonia. We often talk in the foundation around, like, we need to find really smart researchers and pull them into the foundation, pull them into the field of dystonia. And so it sounds like that, that's, getting you involved. And specifically, when it came to identifying this gene, what role did the DMRF play?
Well, first of all, they funded the whole thing because the NIH just wasn't interested. They said, it can't be done. So they helped us find the families. And, you know, like I said, it went on for years, and every year we would go and report our findings and, you know, receive immense support from the foundation, just emotional support that, you know, we need you and, you know, don't give up kind of thing. I think that was
Also, the DMRF funded Stan Fon's group as a center of excellence for dystonia, you know, and so a lot of the clinicians that you see, you know, today who are dystonia clinicians got their training through that center of excellence. There was one in New York, and there was one in Calgary, I think. No, British Columbia, someplace, Donald Collin. And those two centers, you know, basically put the clinical features of dystonia on the map, because they, you know, really studied dystonia. And like I said, the only way that we could find this gene was to have families, you know, because as Dr. Patel said, dystonia is very heterogeneous. You don't, there's, you know, tons of different genes for dystonias. The only way to find the one gene was to have clinicians that really understood what the clinical features were. And so.
Yeah. So we,
I would also like to say, you know, there was an amazing coincidence that, you know, Malin DeLong, who was also supported very strongly and led the Dystonia Medical Research Foundation, he's the one that really discovered deep brain stimulation and pursued its role in helping dystonia patients. So he took the, you know, and he, knowing the information of what genes were causing dystonia, helped to identify that, you know, the DYT1 patients would be one of the most responsive. So
to DBS, yeah. So we do have a few photos from that time. So I'd like to ask Veronica if she can put those up, and you know, talk to us a little bit about like what's going on in these photos. You know, take us back to the time when these photos were taken.
I'm not sure exactly where the photo was taken, but there was Jeff Hewitt, who worked with us most of the time. He was a technician, very dedicated, very competent. The other man there is what's his
Tom
name?
Gasser.
Yeah, he was from Germany, actually. He came and helped with the project, went on to clone Parkinson's disease genes in Germany, and actually we gave him a lot of the samples that he used to do that. And then there's Lori, and then this is
That's Dennis
Dennis
Kessler.
Kessler, when we had the celebration after we
When we actually found the gene, yep.
Yep. And it's not clear in this picture, but more in this one. This is the only time that Lori has ever worn makeup. Dennis insisted.
And we had to do some TV interview or something.
Right.
We had to do a TV interview, and they said we had to do that, so.
Yeah, and that's another one of the donors at the celebration. Who is that, Lori?
I think that's Mr. Fasciano.
His son turned out to have, I think, DYT6, right?
I think.
But it was definitely overlapping the phenotype. And this, I forget his name, but he really was a major player at the end, because I think everybody had just about given up hope at this point. And we needed a sequencer. And in those days, you did your sequencing in your lab. You didn't send it off as we do now to a sequencer. And he donated the money for that sequencer.
Yeah. Well, thank you for going through those. So, you know, I guess moving on a little bit to kind of the clinical side for Dr. Patel, you know, how does having a test for DYT1 help when you're helping, you know, working directly with patients? You know, what, like, walk through, like, how would you use a test for DYT1?
Yeah, so as was discussed, you know, one, dystonia is a very different disorder. So every person looks a little different. The history of it, even before this was recognized as a condition, you know, in the 1800s and early 1900s, a lot of people thought that this was psychogenic, something that, you know, was a manifestation of anxiety or depression. So recognizing that there was a genetic disorder and there's an abnormality in a gene really kind of changed perspectives of physicians and those that don't have as much experience. You know, I was fortunate to have trained with somebody who trained with Dr. Fon and his, you know, Dystonia Center of Excellence. So I got to see a lot of patients with dystonia and, you know, pick up the subtle features and become kind of refine my eye and my diagnosis. But a lot of other physicians may never meet a patient with dystonia, or if they do, they don't even know what it is, because we spend maybe a sentence or a paragraph in our medical school training on this condition. So to have a gene and be able to identify this is the cause really kind of, I think, brings that out to making the correct diagnosis and maybe getting over to the right center for care. And then for me personally, when I'm taking care of a patient, there's a lot of different therapies to offer. And currently, and there's many other genes, but there are, I think, 28 or 29 DYT genes recognized. And there's a lot of people who have been gene tested and we don't even recognize the gene yet. So there's still more to be discovered. But one thing that really kind of came to light with recognizing DYT1 and all the work that went in after recognizing it is this gene in particular is exquisitely responsive to deep brain stimulation. And so when we identify this gene, I'm more likely to say, okay, I'd rather move you through to deep brain stimulation sooner rather than later. Whereas a patient who may have a different gene that isn't as responsive or a gene that isn't recognized, I'm going to spend a little bit more time with botulinum toxin therapy and a few other interventions before we jump straight into deep brain stimulation, because the results are a little bit more varied. So this really helps kind of focus and steer treatment. And then the other part of this is family planning. And so some people who, unfortunately, it's an expensive process, but some people are able to do pre-implantation genetics and screen out this gene so that the next generation doesn't have to suffer. And I think that's a really huge, valuable tool for just recognizing this genetic condition and being able to kind of plan the next generation.
It was, that specifically was incredibly powerful for my family. It wasn't until I actually, started seeing a physician at Rush for my dystonia that this was even brought up to me as an option. So like I had actually kind of, said like, hey, maybe having kids wasn't for me because I didn't want to, be a part of, continuing with dystonia, and I would just maybe adopt or do something else. But having a, family planning, having this brought up as like, hey, you can go through this process, and it's painful, it's expensive, it's, not fun, but it is remarkable that we have the ability to do this. And it is because of the discovery of the gene that we're able to, specifically for those with DYT1, we can do these things to make sure that you can have your biological children have a, high chance of not passing it on. So I was very, very happy to hear that. And we did go through that, as I mentioned, we have two children now through that process. So I kind of want to talk about, we said the words penetrance earlier, and we defined that a little bit. But for Dr. Zulius, so my understanding so DYT1 is always described to me when we were going through this is like, this is an what they say is autosomal dominant, which I replaced in my head as 50-50 chance that the parent passes it on. But when so say you had the gene, we've always been told like dystonia penetrance for DYT1 is about 30%. So if you have the gene, 3 of 10 people would actually present, actual like symptoms of dystonia. One is that do what I say was accurate there, and is that normal penetrance for a genetic disorder?
So what you said is accurate. So penetrance, usually when we talk about penetrance, we're mainly referring to autosomal dominant diseases. So as you said, diseases where you only need one. So we have usually for every gene except for on X and Y chromosome, we have 2 copies, one from mom and one from dad. And so autosomal dominant means you only have one bad copy and you can get the disease. All right. And so penetrance is you can carry the disease gene but not express the clinical features. And so that's reduced penetrance. And so you can have the reduced penetrance can be anywhere from 90% down to 1%. And so it just depends on the disease and how it works. Basically, the idea about penetrance is that there's something else that's moderating. And so that could be other genetic factors, or it could be something in the environment that somehow moderates the expression of the disease gene. All right. So you could have factors that protect you from expressing, and you could have factors that help you to express the disease gene. And so like, for instance, if we could figure out, are there protective factors that stop individuals from expressing the DYT1 gene, can we then exploit those to come up with some kind of a treatment? And so we haven't found, well, we have found one thing that is related to penetrance of DYT1, which is another variant within the DYT1 gene. It's called D216H. And so if you have the H allele at that one variant that's within the Dystonia gene, the DYT1 gene itself, then your penetrance goes from 30% down to 3%. So you're protected. And we don't really know the mechanism of that. We think it's somehow related to how the protein folds or moves or whatever, but we don't really know the mechanism. But we do know that if you have that variant, then your likelihood of expressing the trait goes down even lower than the 30%. It goes down to like 3%. You know, like other diseases, for instance, Huntington's disease is also autosomal dominant, and it shows what's called an age-dependent penetrance. So Huntington's disease, usually the age of onset is in the 40s, like 45 is the average age of onset. And if you have the Huntington's disease gene with a mutation, if you reach the age of like 60 or 65, then the penetrance goes up to about 100%. But if you can have reduced penetrance before that. And so penetrance can mean different things in different diseases. I don't know if I helped or not, but.
No, I personally found that fascinating as someone who has DYT1, and I was actually not aware of that the allele that potentially could be linked to reduced penetrance. So that's interesting. An interesting field of study. Moving on a little bit, Dr. Brickfield, I wanted to ask you a question on how has the identification of the DYT1 gene affected dystonia research more broadly?
Well, I think actually Dr. Patel kind of highlighted that. Remember, we couldn't get funding because there were so many different types of dystonia that the National Institutes of Health was like, how are you going to pull out one? And Lori explained very clearly, was this excellent clinical team that could find the people that had the same type of dystonia and was distinctive. But I think once we found one dystonia gene, people began to realize you just have to be very rigorous about defining the phenotype and getting enough, patients with that phenotype. But I think we're Dr. Patel was saying there's probably like 28 genes now that have been identified. Lori would know exactly. So, it just gave people the realization that this isn't hopeless. I mean, you can sort this out and find individual genes, and then you can make better predictions about the course of the disease and, potentially better ideas about what kind of therapy might work.
I think
Thanks.
The other thing that discovering this gene did was bring more people into the field. We were kind of a lone station for a while there. And I think once it was shown that there is a gene for dystonia, and you can find them, then more people just said, well, let's, work on this. And also just once you find a gene, then you have a protein and people start to think about like, what does that protein do? And how can they, think about working on that protein? And so it just brings a lot of different people into the field, and that helps dystonia research in general, because you have a lot more people, thinking about it and working on it and trying to work together to solve it.
So
Yeah, because we discovered a new protein, we had the opportunity of naming it. And I have to say that I wanted to name it Lauren after Lori, because she had done most of the work, but she didn't want to have a disease named after her. So we spent a lot of time to pick Torsin as the name of the protein.
Yeah. So, kind of on the note of like identifying other genetically inherited dystonias. And so DYT1 is why we're here tonight, and mainly to discuss, but we did have an audience question around what is DYT1? Why is DYT6? And we'll go to you, Dr. Azulis, because I know that you were involved in this pretty closely. So can you tell us what is DYT6?
So all the DYTs refer to, before we find the gene, it refers to like a locus. It refers to that positional cloning place where we put it, right? And so DYT6 is another gene. It's located on chromosome 8. DYT1 is located on chromosome 9. And basically it defines another genetic form of dystonia. But the clinical presentation of DYT6 is overlapping. So Dr. Patel can probably tell us more than I, but is overlapping with DYT1. The onset's maybe a little bit later than DYT1. Generally, the onset is more in the arm rather than in the leg, as opposed to DYT1. And there's a lot more vocal, craniofacial, speech swallow stuff in DYT6 on average. Like Dr. Patel said, every patient is different, but if you kind of look across the average of who has a DYT6 mutation, these are the things that kind of fall out. And so with DYT6, we had a bit of a clue or a bit of help to try to find this, because there was a very large Mennonite family, like 6 or 8 generations that were identified who had dystonia, in this family. And so we could use that family to try to find the gene. And that's actually how we found the DYT6 gene, was using this very large Mennonite family. And then after we found it, we looked for other families who had similar phenotype, similar clinical features, and tested those. And then we found that they also had mutations in the same gene.
I'll repeat my comment about it. It sounds just like science fiction to me, and I appreciate it very much. So I'm going to move on to a question that I'll start with you, Dr. Patel. This is really for the whole group, though. If anybody wants to add in. So if a person has a focal dystonia, like cervical dystonia, why should they pay attention to DYT1 research?
That's a great question. So as we talked about the heterogeneous nature of the presentations, DYT1 certainly could manifest as just pure cervical dystonia. It typically doesn't remain that way. I anticipate or expect some of my patients to progress to develop dystonia in other areas. But cervical dystonia it could be caused by any number of genes. And for many of my patients who have gene tested, they don't come back with a positive gene. So I think it's a opportunity to stay engaged in research, because we may be able to figure out what else to do, learn more about the condition, maybe find that unifying gene or a unifying kind of cause. And I think that's the reason to stay engaged, not only with cervical dystonia, but frankly, any of the dystonias, especially if you had gene testing and it was negative. I tell a lot of my patients, don't get discouraged. There's a lot of people who are studying this now, and we may next year have another DYT29 or 30. So just to kind of stick with it, and this informs us on how to take care of other things better.
Thanks. Anything to add? So I guess on that note, kind of so for the folks who, want to participate, right? So I mentioned early on, I was very young, so this was not my personal choice, but my family did participate in this, and there was, blood drawn, things like that. I'm very happy that we did it, obviously. But if you want to participate in a study, like, what's the best way to get involved in that? And I guess maybe we'll start with you, Dr. Brakefield.
I think you should probably start with Lori, because actually I've kind of switched from finding dystonia genes to trying to figure out gene therapies actually. So
okay, yeah, we'll start with you, Dr. Zulis, then.
Well, I think a lot of studies are now listed on things like clinicaltrials.gov. So you don't have to be just a clinical trial to go on clinicaltrials.gov. You can be, any kind of research that people are doing. And so that's one resource people can look at. Another resource would be talking to the DMRF, because oftentimes researchers talk to the DMRF and say, like, we're doing this study, can you advertise for us in your newsletter? Also, the Dystonia Coalition is, doing research throughout the country with a bunch of different centers. And for instance, I'll talk to the Dystonia Coalition and say, like, we are looking for patients with X, Y, and Z phenotype. And they'll say, yeah, we've recruited this many and you can use them through us or what have you. There's also dystonia registries. probably someone from the foundation can speak about this a little bit more, but there's like a global dystonia registry, and there's a DYT1 specific registry. And so that allows people to get their names on things that then say that they're potentially interested in research. And so somebody like me, I can go to the DMRF or whoever runs these different registries and say, like, I'm interested in this. Can you put something out through the registry that tells people that we're doing this kind of research? And you can also talk to your own neurologists and ask them what they know about studies that are going on.
That's great. And thank you once again also for mentioning the foundation. You know, you can always call or email the foundation. We have folks who are happy to get back to you as well. So, you know, moving on a little bit, you know, since the discovery of DYT1, we kind of, you know, talked about like what just more general dystonia research. Any, you know, is there any like research that like what kind of flew from DYT1? Like we got this gene, we identified torsion. What can we do with it? Like can you talk a little bit about that research and anything that you're aware of?
Well, I think a lot of people have tried to figure out what this protein does. And it does a lot of things. That's part of its problem. It's really a master of all trades. So it's important in, you know, processing of proteins, in terms of cell migration, in terms of different metabolic aspects. Why the patients are just primarily affected in the nervous system is not clear, because it's expressed in all cells of the body. So we spent many years trying to figure out exactly what the gene does in the hopes that we could find some kind of drug therapy, but we really just found out that it did a lot of different things. And I know that there's another group called Tyler's Hope, and they are trying to develop a drug to treat the DYT1 dystonia, and they're pretty optimistic about it, but it just remains to be seen. You know, you have to do clinical trials, and so people, are trying to, and there's still people, you know, figuring out the structure of the protein, you know, et cetera, et cetera. So there's a lot of work that just goes on for that protein and for other proteins that are involved in the dystonias. I think, like I said, primarily in the hope of finding a drug that would work.
And there's there's also animal models that have been developed. And animal models are an important step in terms of something to test the drugs in. And so usually when you find a disease gene, one of the first things that people do is try to make an animal model, a mouse model, so that they can then do a lot more experiments in the mouse and try to understand what the disease process might be in the mouse. And again, if there's any kind of treatments, then you try the treatment on the mouse first. If you have some kind of phenotype that you are, you know, as associated with the disease gene, and then you see if you can correct it. And people are also doing that now in what we call iPSC cells. So those are induced pluripotent stem cells, where you take a fibroblast from a patient and then you can differentiate it into neurons or glia or other kinds of cells. And then you can, you know, again, test certain drugs or things like that. And there's also like fly models and worm models. So there's all kinds of different model organisms people are using, all of which are basically to hopefully be able to test, find phenotypes that you can then rescue with some kind of drug or some kind of treatment.
Yeah, I would say the frustrating thing about DYT1 in that regard was people made animal models and they didn't have dystonia because the human brain is just different than the mouse brain, and is
right
in that particular. So we're never able to get a really authentic mouse model for DYT1, but I think for some of the other dystonias, they have good models.
And when you say they didn't have dystonia, like they would have a mutation, but they just wouldn't show symptoms.
Didn't show any symptoms, yeah, right. We tried stressing them, you know, we tried trying to find other factors that would induce it, but it's just the human brain is put together a little differently than the mouse brain.
Yeah. So we had a question around a person who had a maternal grandmother who had dystonia, but they've themselves have been tested, and there was, you know, no identified dystonia gene. how likely is it that the dystonia would, because we have non-genetic dystonias, how likely is it that it was, like how likely was it that it was dystonia that is genetically inherited versus some of the other dystonias? Maybe Dr. Patel, that's a good question.
Yeah, I was going to say, it sounds like a question I could try to answer. So
Yeah.
It is one of the challenges and the frustration of predicting the future. We as doctors are not very good. And even if you test positive for DYT1, there is a small but real chance you may never get it in your lifetime. So what I tell my patients who have family members who have dystonia or other neurological conditions is just take care of yourself. Exercise, eat well, do all the right things and take care of yourself. And if it manifests itself, we will take care of it in the best way that we know how at the time. And with the rate at which things are advancing and the more that we're learning, we may have more genes to test than we do now. And one of the things to talk about is, we mentioned a few times that there's 28 genes recognized for DYT1. A lot of these are developed in labs and they're not commercially available. So most of the gene panels that we run, and like when you come see me and I say, hey, I think you have dystonia, let's try to run a panel. It's an incomplete panel. We're probably testing somewhere around 6 to 8 of the most common genes, after which we can't really recognize all the ones that aren't on that commercially available panel. So we are missing a lot just on that clinical forefront. And it's the kind of the small group researchers that are looking for very specific patterns that are studying this and then kind of helping it to become more commercialized.
So we had a question from someone who has a rapid onset Parkinson's dystonia, and they have been tested and they don't have the DYT1 gene, but they have family members that have cervical dystonia and tremor. Should they be concerned about their kids developing dystonia? Probably again for you, Dr. Patel.
Yes, but concern is we could be preoccupied with worry and have nothing to be able to control. So I think the most important thing is to stay connected with the medical community, especially the research community. And I'll circle back to that in 1 minute, which is as things change and develop, their kids may have a better opportunity to identify that gene. And then hopefully we can we can offer a treatment intervention. But I kind of tell my patients, we can't worry about something we can't control and we can't predict. So I tell everybody, go forward, live the best life, stay connected. And one thing about the Dystonia Coalition projects, which I'm involved with at our center and have been involved with in my career, is we are looking for families and patients with specific symptoms that we then collect samples and we collect clinical data so that we can help identify what the cause is, and we can try to help recognize, pull that samples with all the other patients. So being part of registries and being part of, especially the Dystonia Coalition, because we're not looking for genetic causes, we're looking for patients who have symptoms that haven't been diagnosed. I think will be the kind of that next step to answering those next questions.
Got it. We also had a question around the relationship between dystonia and other health issues like IBD and things that have high prevalence in the Ashkenazi Jewish population. Dr. Azulia, this is maybe a good one for you to start with. Like, is there a connection between these?
I'm not sure there's enough research to know if there's a connection. Basically you kind of have to look at the whole population and ask, do you see, dystonia is relatively rare in the population, whereas IBD is more common. And so you have to kind of look at the dystonia population and ask, how often do you see IBD? And is it more often than you would expect by chance based on what the prevalence of IBD is in the population? And I don't think that those studies have been done. So it's hard to answer that. I was glancing at that's why I'm sort of looking off to the side. I was glancing at the questions, and somebody asked about something about Ashkenazi Jews. And so the reason that we sometimes use Ashkenazi Jews is because they're more of an isolated population where they tend to marry within their group, sort of like the Mennonites. They tend to marry within their group, and that allows you, so they have what we would call less complexity. We're talking about heterogeneity. They're more homogeneous, and therefore it allows you to find genes in that population because there's less genes to look for. There's less different genes to look for within that kind of more homogeneous population. And so that's why a lot of people concentrate on these smaller populations like Ashkenazi Jews or Mennonites or Amish or French Canadian or what have you.
That's actually something I hadn't quite realized. That makes a lot of, well, when you think about it, that makes a lot more sense. We had a question around, folks wanting more information around family planning to eliminate dystonia. And, I think Dr. Patel, maybe we can go to you and you can kind of say, give a little bit of a blurb about what you would tell people, how maybe start the process. I'd also make a pitch for reaching out to the office, the Dystonia Medical Research Foundation office, because we can connect folks with various centers, reproductive centers that can help out with this.
Absolutely. So I think one is identifying what gene you have, whether it's DYT1 or something else. And then honestly, I do reach out to the foundation for patients who are looking, because a lot of times people are traveling a great distance. And even though the expert might be at a specific center, it doesn't, the resources for family planning aren't always there. So I think that would probably be the way I would recommend it. At Rush, we do have family planning options, but at my previous job, we did not. So I often was directing patients to the foundation if they wanted to pursue that any further.
Got it. We had I think, Dr. Zulish, you're looking through the chat. We had additional questions on kind of what factors affect that penetrance, and, maybe just go over the part about the allele again. I know you said this is the one thing we've seen that has dropped it from 30 to 3. I guess maybe kind of talk a little bit about is there hope there to find out more of these things? How would you use, if we found out more of these, how would we potentially use them?
So again, there's potentially either other genetic factors or environmental factors that could influence penetrance. And so, environmental factors, because I'm a geneticist, I guess, environmental factors are harder to look at because, so there's been studies to look, for instance, at head trauma. Is head trauma associated with manifesting more often DYT1 than not? Or is there's also been childhood illnesses like chickenpox and those kind of things looked at, before vaccinations and after vaccinations kind of deal. But it's a little bit hard to look at those environmental things because most of the time the patient that you're talking to is 20, 25, 30, and you're asking them about something that happened probably that they don't, yet they actually have to ask their parents about, probably, right? Did I ever fall off the swing or hit my head or what happened to me, And so that's why, and of course, when something happens and then you get a bad disease, you're going to remember that more than when something happens and you don't get the disease, right? And so that's part of the problem with environmental studies, is that there's a lot of recall bias and not even being able to remember things and trying to document things that are from a long time ago. And so, but in terms of genetics, the question is, so for instance, if we find torsion, we can ask, once we know some of the functions of torsion, we can ask, what are the genes that are the proteins that interact with torsion? And is it possible that some of those proteins that interact with torsion then have variants in them that either make torsion work better or make torsion work worse? And those potentially could be things that would influence penetrance. And so those are the kind of studies that you're thinking about, is what is it that, and it could be something totally unrelated to what we know about torsion also, which may very well be the case. But, basically to do this kind of thing, you get a group of people who are affected, And then you get their siblings or their relatives who, what we call non-manifesting carriers. So these are people who have the gene but don't have any of the clinical symptoms. And you basically ask, what's the difference between them? So you go again through the whole genome and you look at their genome and you say, what do the affected people have other than Torsin A that the unaffected people don't have, right? Because they both have Torsin A, but there's got to be something different about them genetically, we think. And so that's basically how you do the study. But, we don't have huge numbers. We don't have like 50,000 people who are clinically affected and 50,000 people who are non-manifestors. We have maybe 200, and so then it becomes a game of, numbers and statistics and that kind of thing. And so it's a little bit tricky to find things.
So I think that I will say like that does cue up a question that I saw which was sort of, what is torsion? We've used the word here a little bit. I mean, as a layperson who's not trained as a scientist, I know it's a protein, but I guess can you maybe just give us a little bit of an understanding to the best of your ability?
John,
Well,
do you want to do it?
It's a protein that's expressed in all cells of the body. If you know the structure of a cell, there's the nucleus. It's surrounded by a nuclear membrane, and then that flows into what's called, I hate to do this to you, endoplasmic reticulum. So when proteins are made, if they're going to be secreted, for instance, they have to go, the RNA that they're translated from, so the RNA for, let's say, torsin, and for many other proteins, gets funneled into the endoplasmic reticulum, and it helps to fold other proteins into the correct structures as they come through. But then torsin also aligns itself with, I would call them like cytoskeletal proteins within the cell. So for instance, when a cell is migrating, it actually uses torsin that's more around the nucleus to help the cells migrate. So it's a kind of protein that affects the ability of the cell to make other correct proteins and also to move. And I don't know if Lori wants to add anything to that, but you probably could. It's supposed to be involved also in protection from stress if the cells are stressed for some reason by any kind of stress, like temperature, something like that.
The only thing that I'll say is to back up slightly in that the DYT1 gene that we found encodes torsin. So the DYT1 gene, the DNA, when you translate it into RNA and then the RNA becomes protein, becomes torsin. So torsin is the same, the product of the DYT1 gene. So Zander gave you what we think torsin does, but I'm just telling you that's how DYT1 gene is related to torsin.
Right, and because it has the mutation that causes DYT1 dystonia, causes the removal of an amino acid, the protein, the torsin itself folds slightly differently, so it kind of compromises its function. It's quite unbelievable how this little change in a protein could cause such a disease, but there you are.
Yes, such a kind of monumental change in the macro environment.
Mm-hmm.
This is a good question for you, Dr. Patel, potentially, or we can also pass it around. Is do we know what percentage of people who are affected with dystonia have a genetic form of dystonia?
Oh, off the top of my head, I don't know that I can answer that accurately. I don't know if either of my colleagues have that number. I can speak to it in general terms, but that's about the best I could answer. Do you guys know?
Lori may have some idea.
Well, what percentage of dystonia patients have a genetic form? So this is a tricky question because, again, I'm a geneticist. So I think everything's genetic. But so, for instance, focal dystonias, we don't really have good genes for focal dystonias, and about 90% of dystonia cases are focal dystonias or segmental. So that's, your torticollis, your blepharospasm, your spasmodic dysphonia, your writer's cramp. So it's possible, and most of those focal dystonias are thought to be sporadic, where people don't have a family history, but it's about anywhere from 12 to 25% of people with a focal dystonia do have a family history. And it's thought that the penetrance in focal dystonias is even less. It's only like 10 to 15% penetrant. And so I would say that there's possibly risk alleles that increase your risk of having a focal dystonia. So it may be, unlike having DYT1, where you get the gene and you potentially get the disease. Again, it's only 30% penetrant, but it may be that there's a combination of different variants in different genes that come together that push you over some level that then allows you to express a focal dystonia. And the reason that it might be hard for us to find these is that it may be a bunch of different combinations. So, torticollis patient 1 may have 3 different variants that push them over to expressing their trait, but torticollis patient 2 has 3 other variants and totally different genes. And so when you're looking at all the torticollis patients together, again, we're get this heterogeneous population, we don't have enough power to pick out the small groups. And so that's when you use things like imaging, for instance, and you can ask, is there a difference when you look at imaging of torticollis patients where you have a group that has, some imaging difference than another group? And then you can say, well, maybe then I can group this group of torticollis patients who clinically look exactly alike, but they have what we would call an endophenotype, a subphenotype that may allow us to put them into a different group that would then allow us to say, okay, let's see if we can find what's genetically going on in them. And so even though there's the vast majority of focal dystonia patients look like they have sporadic disease, I would say there may be risk factor genes, just like when you think of like heart disease or cancer or whatever, that there's risk factors that increase your risk of getting something, but don't tell you 100% you're going to get that.
Yeah.
I don't mean to scare anybody. I'm just
No, yeah. Go
I
ahead,
would,
Dr. Bell.
in kind of practical terms for, I see patients and I kind of advise some people to get gene testing and some people not. And that's why I said I could probably speak to it in general terms. When I see somebody who has kind of most of their body affected, what we call generalized dystonia, that tends to have more of a genetic predisposition than the focal dystonia where just your head is turning or just, you get a writer's cramp or there's a hand or a leg dystonia. So from a pragmatic standpoint as a physician, I would love for every single one of my patients to get gene tested, but it is not always covered by insurance and it can sometimes be a pricey endeavor. So I kind of pick and choose my battles with the insurance company. And I tend to recommend my generalized dystonia patients get gene tested, and then my focal dystonia patients if they want to. But I don't generally recommend that every cervical dystonia patient and every limb dystonia patient that has nothing else and no family history get that gene tested. That kind of helps.
Yes, I agree with that. I agree totally with that. In terms of practicality of patients, the early onset generalized folks are the ones that are going to have genes and that we know of, or genes that have been identified so far. The focal dystonia guys, less likely to have genes that we've identified so far.
Yeah. So in the interest of time, I have one last question for you, Dr. Brickfield, and that was, is there hope to be able to remove the dystonia causing gene in people who are already symptomatic?
That's a very good question. We could, with gene editing now, there's the potential, but it's a long way in the future, I think, to actually correct that mutation in cells in the body. But what percentage of cells you could correct it in, and which cells you would want to, somewhere in the brain you'd want to correct those cells. So in the future, it's possible that could be corrected. And just as people who have suffered from very severe dystonia, generalized dystonia, and gotten deep brain stimulation, have shown immense normalization. It's definitely reversible. So I think if you ways are being developed to try to correct genes in people, and I think DYT1 is a good candidate because I don't think that many, cells you would have to hit. And because you're going anyway, usually with deep brain stimulation, you'd have the opportunity to introduce these correcting proteins in. There was another good question I saw, but we're getting late here. So, but I would like to answer. Can I answer one more question I saw on the screen?
Yeah, you can.
very good
Yeah,
question.
we go ahead. And I do want to also add that, for questions we weren't able to get to, we do really appreciate them. And the foundation is collecting them and will be attempting to get answers out to the people. But yeah, no, if you
Yeah.
spotted a question you wanted to highlight, please do.
Yeah, I mean, someone asked, is this deletion, is three base pair deletion the only thing that causes DYT1 dystonia, or could there be other mutations in that gene? And that to me is one of the most fascinating things, and it seems to be very unique. Other mutations in the gene in the heterozygous state, as what I was explaining, you have a normal gene, now you have a DYT1 gene that has a different mutation, they're normal. It's only if that person or their children gets two different mutations in that gene and two copies of them, then they have a severe developmental disability. I mean, it just totally messes up the development of the brain. So there's something very unique about this three base pair deletion.
And
So no, go ahead.
I am also seeing a bunch of questions about people who want to participate in research and looks like they have more than one person affected in their family. And, so I'm happy you can give them my email address and I'm happy to talk to them because we're always looking for families for research. So.
Yeah, that's great to know. Thank you. So I and do want to kind of close this out here because we have hit our time. So first I want to thank each of you for joining me here tonight. I really deeply appreciate it. And I appreciate the work that each of you are doing in this field. And for those who are, tuning in to participate, the theme that we wanted to highlight here tonight is that although we know science is moving slowly, there is reason to be hopeful. And progress takes time, but it does require participation, so for those who are affected, who want to help with studies, there are multiple ways we talked about in the session to do that. But again, thank you again for joining us. We really appreciate it. Thanks.
Thank you for
Right.
having us.